Great, thank you very much, and good afternoon, everyone. Delighted to be able to talk to you today about Albumedix and the use of recombinant human albumin within advanced therapies. But first, just like to mention, as stated here, we are now a Sartorius company. It was closed at the end of September, and it's going to be super exciting to be part of Sartorius, accelerate the growth of the use of our recombinant albumin in the advanced therapies, and also develop new products and new ways to help the advanced therapy field with our new colleagues. So, a bit about Albumedix. So we've been producing recombinant human albumin in its current form for over 25 years, been actually in the business for over 35 years. And we have a GMP manufacturing facility in Nottingham in the UK. It's been regularly inspected by MHRA, also inspected by other leading authorities. And we produce our recombinant albumin in Saccharomyces cerevisiae, a baker's yeast, so it's completely devoid of any human or animal components. It is the world's only USP NF compliant recombinant albumin, and all of our products and all of our albumins are backed by drug master files. As I say here, we like to sort of consider ourselves a partner to our customers. We're not just a supplier of albumin. We want to help our customers in order to best use our albumin, help them through any regulations, quality aspects and things like that, so that they can get their life-saving therapies to the market as fast as possible. As mentioned, we've been in the field and producing albumin for a long time. Our longest serving customer is Merck, who use it in the MMR and Procod vaccines and have used it since 2006. And since then, it's had over 200 million pediatric vaccinations, probably a lot more than that now, because that number's a few years old. But overall, I mean, that just sort of emphasizes the safety profile of our recombinant albumins. So our business is based on sort of three pillars. Our recombinant human albumins, Recombumin, which I'll come on to. Veltis, our drug delivery sort of technology, where we've used our knowledge of albumin to develop albumin variants to improve things like half-life or drug loading. And then enhanced services, which we've always sort of been doing, but sort of launched a bit more formally, sort of more recently, where again we're using our knowledge of albumin to help our customers to either sort of develop sort of formulations using our albumin for viral vectors, cell therapies, or develop albumin formulations specific for media types and for specific cell types. But also to provide regulatory and quality support to help guide our customers through any regulations about the use of our products in their hands, to get their product to market as soon as possible. And then in terms of Recombumin, we have two products, Recombumin Prime and Recombumin Elite. They are both extremely pure, both have very good batch consistency, no human and animal-derived components. And the main difference is around the formulation, with Recombumin Prime, sort of our initial product, formulated very much like clinical-grade HSA, whereas Recombumin Elite was developed for cell therapies and as a simpler formulation. So albumin is a wonderful molecule. It can be used in a broad range of different applications, and we have applications around sort of protein peptide formulations, small molecule solubilization, been a lot recently in terms of medical devices as a coating to the surface to make them biocompatible, Diagnostic reagents, we've seen an increased amount, but obviously relevant to today and such like is in the advanced therapies, so cell therapies, gene therapies, and viral vaccines. And as I say, we have customers in all of these areas. In terms of the cell therapy, you can sort of map out a sort of a process. I mean, it's very generic and very different for different places, for different therapies. But we can see a use of albumin across the board, right into the medias for things like expansion, differentiation of cells, through to any separations, any processing of the cells, and then through to sort of formulation, dose prep, and things like that. In the media, what we're seeing increasingly, I mean, it's across the board really, is that not all albumins are the same. You get different performance benefits with different albumins, etc. And a lot of that is down to the purity of the albumin and very specifically what's attached to the albumin. Albumin as a carrier molecule can carry both good and bad ligands and such like. And what we pride ourselves in is that we have what we believe is the purest albumin out there on the market in terms of lots of things, and especially in terms of ligands, so that we can then manipulate that and modify that depending on your application. So today, though, I'm going to sort of concentrate on the back end around and just give you a few case studies for the sort of the cryopreservation and formulation. And the first one is with mesenchymal stem cells. We did this in collaboration with Excelia in Barcelona, who have been developing some therapies, to say, with MSCs. And they had a protocol that used HSA for the cryopreservation and thawing of their therapies. But they were seeing that they could only really have a shelf life post-cryopreservation of about 12 hours because after 24 hours, as you can see here in the black graph, black lines, is that it was below their viability specification that they'd set. What we did is we replaced the serum-derived albumin with our recombinant albumin, our Recombumin Elite product, and here it greatly improved the stability of the cells, meaning that the viability was maintained above this threshold for over 72 hours, which greatly improved the sort of logistics, could help deliver the therapy to the patients far easier. We obviously monitored the potency and identity of the cells, and yes, they had the correct cell markers, and they could differentiate into the correct cell types, etc. So we have published that, and the link is on the presentation. Secondly, with induced pluripotent stem cells, this is again with someone in Barcelona, and here we were looking at the use of albumin in various different media types, so plus or minus albumin, and looking at whether or not the iPSCs could then grow on post-cryopreservation. And as you could see, the inclusion of Recombumin Elite into all of the different medias greatly improved the ability of the iPSC cells to generate colonies. That's not surprising for things like the standard isotonic solutions and MT cell, because they're not designed for freezing of cells. But even with a design media for the cryopreservation of induced pluripotent cells, then you could greatly increase the number of colonies by the addition of Recombumin Elite. And it wasn't just the number of colonies, but also the size of the colonies as well. And again, there was no change in the pluripotency state. In-house, we've done some tests with T cells. This is looking at total viable cells, total live cells, post-cryopreservation, looking at the addition of different albumins or no albumin at all. And again, you can see that with the addition of Recombumin Elite, you could greatly increase the number of viable cells even after 48 hours using Recombumin Elite compared to the other albumins or no albumin at all. And then finally, on the cell therapy side of things, this is with sort of hypothermic storage at ambient temperature with no cryopreservation. We have a number of customers who are wanting to sort of remove the cryopreservation step, not have any DMSO present at all. And so we looked at that with, say, MSCs, and again, looking at cell viability. And again, here you can see a significant improvement in the stability of the cells with the use of Recombumin Elite compared to other albumins or no albumin at all, such that even after sort of 96 hours at ambient temperature, you still had a very high level of viability. In addition, the actual cell markers were maintained with Recombumin Elite compared to some of the other samples as well. So the Recombumin Elite was not only maintaining viability, but also maintaining the identity of the cells. So just briefly going on to viral vector side of things. So again, you can map it out similarly. And again, we believe albumin can be used throughout the process, both within sort of the upstream side of things in the production of the viral vectors, where we have seen by the addition of albumin into serum-free media, then you can increase the actual infectivity titer of the viral vectors. So it's not just the number of particles, but it's actually the quality of the particles themselves. But obviously, with a recombinant albumin, you're still having that serum-free and non-animal-based media. And also, sort of through the process, to prevent adsorption, aggregation, and things like that. Not got too much data internally that we can show. Most of that's been generated by our customers, but there has been just a couple of things that we can show. The first is with Sanofi, who are producing a vaccine, and they were looking at absorption onto a vial. They were monitoring this colorimetrically using gold nanoparticles, and the greater the color, the greater the absorption of the vaccine onto the vial surface. And as you can see with formulations 2 and 3, that color is much reduced, and they contained, as you'd expect, our albumins as an excipient there. So that sort of greatly reduced the amount of absorption of the vaccine. And then finally, this is a HSV vector-based oncolytic vaccine that has very poor stability, has to be stored at -80 for long-term storage, can't be taken out of that -80 for very long, has a poor freeze-thaw stability, poor 2 to 8 stability. And it was shown that a formulation could be developed with recombinant prime that could greatly increase the stability of this HSV-based vaccine, both from a freeze-thaw perspective, but general thermal stability as well, so increasing the stability at 2 to 8 or at ambient temperature. So just to summarize, then hopefully I've shown very quickly the possibilities of using albumin within the advanced therapy field, no matter whether or not that's in the cell therapy side or the viral vector and viral vaccine based. That can be sort of throughout the process, although what I've shown here very much is the stability and the formulation of both of those sorts of things. As I say, we're here to help our customers, so we'd very much like to discuss the use of albumin with everyone. We do have a table out in the foyer, so happy to come and have a chat to us during the rest of the conference. Thank you for listening.